specialisation, which is essentially what naturalization acts on. The system that I use are floating aquatic plants. These are tiny floating unrooted plants that float on the surfaces of ponds, bogs, lakes, sometimes ditches and puddles. They're extremely common to grow all over the world. They're mostly made up of duckweed, and they sometimes coexist with a few species of liverworts. And they are extremely well suited for experimental work. We can run highly replicated multi-generational experiments, which means that we can test some fundamental questions in ecology and evolution that would be difficult to do with larger organisms. Talking a little bit more about the species, the main one is Lemna minor. This is the common duckweed. I'm sure you've seen it everywhere. If you haven't been looking for it. It's not hard to find. It's an aquatic floating macrophyte with extremely reduced morphology. The fronds are just a couple of millimeters. It consists of a single frond and a single root. It's so small, the flowers are microscopic. However, in the field, it actually almost never flowers. Reproduction is almost exclusively vegetative and asexual, which leads to extremely fast generation times of just 2 to 3 days in ideal eutrophic conditions, and extremely large population sizes. So you have hundreds of thousands or millions of individuals in a single small pond. These traits make it extremely well suited as a model in experimental ecology and evolution. On top of that, it's ubiquitous, it's cosmopolitan, it's easy to find, sample in the field, it's easy to grow in the lab, it's easy to culture, and what's more is that the genome has recently been sequenced. In addition to ecology and evolution, it's a model in ecotoxicology and has a number of industrial applications, including for wastewater remediation, biofuel production, agriculture and aquaculture feed, and is also used as a human food source because it has more protein content per mass than soybeans actually. So that's just one of many duckweed species in the family Lemnaceae. There are 37 broken up into 5 genera. The 4 here are Lemna minor, Lemna trisulca, Sparadella polyrhiza, and Wolffia columbiana. They are very common in temperate Eastern North America and often coexist in the field. So many of the chapters in my thesis will be using these species. This is a conceptual figure that I'd like to kind of bring up at the beginning because I'll refer to it several times throughout my presentation. It's a nice way to kind of group together all the different processes that I'm going to be talking about. And different chapters will look at different combinations of processes. So the main question that I'm addressing is how the community phenotype responds to environmental change. This will be a complicated mixture of ecological, physiological, and evolutionary processes. From population genetics, we know that the 4 main evolutionary processes are mutation, gene flow, genetic drift, and natural selection, right? So genetic diversity will enter into a population through mutation and gene flow, and then it will be structured or removed either systematically through natural selection or stochastically through genetic drift. There are analogous processes in the ecological realm, right? Species diversity enters into a community through dispersal, and then it's structured and removed either through species sorting, another word for competition, or through ecological drift. And then, of course, throughout all of this, there's also phenotypic plasticity. I'll be presenting the 6 chapters of experiments that we've done. The first 3, we're going to be looking at one species in isolation. This is Lemna minor, the common duckweed. So we'll be looking only at physiological and evolutionary processes. Then in the final 3 chapters, we're going to take what we learned in the first 3 and apply it to a multi-species community context. All right, chapter 1, phenotypic plasticity, gene by environment interactions, and the microbiome in Lemna minor. So there has been a lot of recent excitement studying the terrestrial plant microbiome. It's well known that microbes colonize every inch of plant tissue that can have important phenotype and fitness consequences for the host plant. Mycorrhizal fungi and plant growth promoting bacteria can increase plant fitness by increasing nutrient uptake, stress tolerance, and pathogen resistance. So it kind of leads to a sort of consensus, I suppose, that the plant microbiome is a highly specialized, largely mutualistic assemblage of microbes. But there's also, of course, in any microbiome, countless competitors, parasites, and pathogens. So here we're studying the duckweed microbiome. So it's been shown that the duckweed microbiome is a non-random assemblage of at least the bacterial part of the environment, and it closely resembles that of the terrestrial plant epiphytic leaf microbiome. But it's a lot more than just bacteria. Here we have a bunch of deloid rotifers clinging onto the root cap of Lemna minor root. In addition, you will have tons of ciliates, flagellates, fungi, both nitrogen fixing and pathogenic, as well as filamentous algae, diatoms, and of course, lots of bacteria. The main objective of this chapter is to assess the impact of the Lemna minor microbiome on the plant phenotype and fitness. And we do this for several genotypes and over several environments. This is because the effect of the microbiome on plant phenotype and fitness might be mediated through gene by environment interactions. We know well that plant genotypes often differ in their phenotypic response to the environment. We know that the composition of the microbiome may vary among genotypes. And we also know that the microbial assemblage may depend on environmental conditions. And the host phenotype may be microbially mediated. This way, the phenotype and fitness of the host plant will be the effects of the host microbiome, the environment that it's grown in, and whether or not the microbiome is present or not. So to test this, we did a field survey across Montreal. We identified eight natural sites close to the island of Lemna minor, brought them back to the lab. And then first what we did is we sterilized them. We killed the microbiome completely with bleach. We then had a single living frond from each site that we grew up into a clonal population. We split into two, and then on half of this, we reintroduced the microbiome by growing the sterilized fronds back in their natural pond water, surrounded by other fronds that still had the microbiome intact for a number of generations. Then to ensure an equal physiological starting point, we grow them all in a common garden. We break up the clonal colonies into multiple replicate flasks, and then we run our common garden assay. After a couple of generations, we measure fitness and phenotype. And here's what we find. So we have frond area as a function of the four different environments. So yes, there's an effective environment on frond area, but really what I want to point out is the difference between the red bars and the green bars, right? Systematically, the green bars are lower than the red bars. So the green bars is when the microbiome is present, and the red bars is when the microbiome is absent. So systematically across all environments, when the microbiome is present, the plants have smaller fronds. Looking at root length, so there's this huge effect of environment. So going from high to low nutrient environment, roots triple in length, common physiological response, of course. When plants need more nutrients, they'll grow longer roots. But again, I'm going to point you to the difference between the colors in the bars. Systematically, the green bars are lower than the red. So when the microbiome is present, plants have shorter roots. Now looking at fitness, which is essentially population growth rate in an asexual species. So of course, fitness depends on the environment. You give plants more light and nutrients, they grow faster. Surprise, surprise. But looking at the effect of the microbiome, again, across all environments, the green bars are lower than the red. That means that the overall effect of the microbiome on host fitness is a -1. It decreases host fitness. Now looking at the different genotypes. So here we have the 8 different sites from which the plants were sampled and the 4 different environments. So this phenotypic response, the response in fitness and growth rate, is relatively consistent across genotype. However, the magnitudes do vary. This means that there is a gene by environment interaction, which leads us to believe that there is some degree of genetic difference between the sites. However, it is not large enough in order to affect the overall physiological effect on plants or the effect of the microbiome. So what have we learned in chapter 1? That phenotypic plasticity is largely independent of the microbiome, which consists largely of pathogens, parasites, and competitors. The microbiome resulted in smaller fronds, shorter roots, smaller colonies as well, I didn't show that one, and decreased plant fitness. The magnitude of this plasticity varies among genotypes, and it suggests that there are genetic differences among sites. So in chapter 1, we experimentally manipulated plant phenotype in the lab in Lemna minor. In chapter 2, we're going to go to the field and look at phenotypic variation in Lemna minor and ask to what degree is this phenotypic variation due to phenotypic plasticity on one hand or local adaptation on the other. So much of biology is concerned with variation, of course. We're interested in variation in fitness and phenotype, both of which have environmental and genetic components. These are, of course, related because variation in phenotype will surely influence variation in fitness. First, we'll look at variation in phenotype, then we'll switch to fitness. The trait that we're interested in is the root to shoot ratio. And this represents a fundamental trade-off in biomass allocation. It's, of course, a well-studied trait for terrestrial plants. So plants can either invest limited energy and biomass into shoots, which is responsible for light capture, photosynthesis, or they can invest it into roots, which is responsible for nutrient and water uptake. For Lemna minor, everything's easy, right? We measure frond area and root length. They are very easily measurable traits. And in the field, we predict that in environments where light is limiting, plants should grow larger fronds. And then in environments where nutrients are limiting, plants should grow longer roots. So we went to the field. And we looked for populations of Lemna minor. Again, just to summarize, the main objective of this chapter is determine if intraspecific variation in the root to shoot ratio is correlated with natural gradients of light and nutrients, and then to assess if such variation has a genetic basis. So we found 37 sites across Quebec in a 3-week-long duckweed hunt camping trip, from Ottawa over here to Quebec City up to Lac Saint-Jean. In each site, we measured 30 individual plants. We measured frond area, root length. We measured percent shading, light availability, and then water nutrient content, phosphorus and nitrogen, and a slew of other environmental correlates. And here are the results. So first, we have the effects of light on frond area. We see that as there's more and more light, fronds get smaller and smaller. And the effect of root length on phosphorus, when there's more and more nutrients in the water, roots get shorter and shorter. So both of these results line up with our predictions. Relatively simple, it makes intuitive sense, but it's nice to find it in the field. The next question is this phenotypic plasticity or is it local adaptation? The common garden is a common tool in assessing the genetic basis of adaptive traits. In the field, of course, all the variation is a combination of both environmental and genetic components to that variation. But if you grow them all in the same environment, you're removing all of that environmental variation. So all of the variation that remains is genetic. So from the field, we bring back samples from 3 different sites from each site. In the lab, we then break them up into replicates and then grow them in the common garden. We track generations very meticulously. You wait 2 to 3 generations to make sure that maternal effects and carryover effects will start to decay, and then you'll measure plant phenotype and fitness. So here we have a whole ton of variation, right? So this is a frond area on the Y. This is measured in the field. On the X, we have the 37 different sites. So the variation among the different bars is the variation among the different sites. Variation within each bar is the variation among clones within sites. So yeah, there's a ton of variation, but then you take the plants back to the lab, you grow them in the common garden, and you see that the vast majority of that variation among sites disappears. There's only 6% of it that's left. That means that about 94% of that variation in frond area that we saw in the field is due to phenotypic plasticity. It's even more striking for root length. Again, tons of variation in root length in the field, bring them back to the common garden, and there's only 3% of that among site variation remaining. So the vast majority of variation in root length measured in the field is again due to phenotypic plasticity. Now looking at variation in fitness. So like variation in phenotype, it will have environmental and genetic components. So this is from the common garden assay. Again, the 37 different sites, different regions across Quebec, and this is fitness. And again, the variation among the bars is the variation among sites. The variation within a bar is the variation among genotypes within a site. And what is kind of shocking about this is how much variation there is within each site, right? We kind of assume that Lemna minor would be largely clonal. They grow so fast, they don't have sexual reproduction, but we actually see that there's twice as much variation among clones within sites than among sites. So how could this be? Perhaps it's because there are high levels of mutation or immigration. But why are we even interested in variation in fitness? Well, it's related intimately to mean fitness. Of course, mean fitness will be degraded each generation by the introduction of low fitness alleles via immigration and mutation, and then it will be restored each generation via purifying selection. If we remember the ancient fundamental theorem of natural selection by Fisher, it equates the rate of change in mean fitness to the standardized additive genetic variation in fitness. So the genetic variation in fitness is a fundamental concept in evolutionary biology because it gives us an idea about the potential for natural selection to counter evolutionary change, environmental change, and the rate of the evolution of fitness. It also enables us to make predictions about the rates of immigration and mutation. So here we have variation in fitness measured in the common garden. So it's only genetic variation in fitness. We can standardize the variation by dividing it by the square of the mean. And this gives us a value of about 1%. How we interpret this is that fitness is degraded by about 1% each generation and then restored by purifying selection. There are very, very few estimates of SVA in the literature, but they range from about 1 to 10%. So we're in about the right ballpark. Just a little bit of population genetics. In this chapter, we have observed strong phenotypic plasticity in the field and have seen that it's correlated with environmental variables. The majority of this variation is environmental, so plastic, although it did also have a genetic basis. The estimates of genotypic variance in fitness were made based on what I just showed, and therefore we were able to make predictions about the strength of purifying selection. However, the phenotype in the field is not necessarily maintained primarily by purifying selection. As I stated before, it's through largely phenotypic plasticity. The duckweed diversity paradox, right? Kind of like the phytoplankton paradox. So how is it so that there's so much intraspecific genetic diversity in a single site? Normally in a population, genetic diversity is controlled by a bunch of factors, right? It enters the population through gene flow and mutation, recombination, at meiosis. It's removed through selection and genetic drift. But in Lemna minor, well, there's no recombination, it's asexual. And it's assumed that there's a severe seasonal genetic bottleneck. Why is this? Winter. So Lemna minor doesn't have any specific overwintering structure like many of its closely related species do. So it's assumed that many of the fronds die off in the winter as it's caught in the ice. They sink to the bottom. In the springtime, a few lucky fronds survive. They avoid freezing if it's deep enough, and they'll float back up to the surface. And then because of the rapid exponential growth, they will rapidly increase until they cover the surface of the pond. And that next year's population will be made up of mostly just a few genotypes, right? They're mostly clonal. But that's not what we found. So the purpose of this next chapter was to assess the severity of this seasonal genetic bottleneck. We did this in a small little pond near the summit of Mount Saint-Hilaire, which is a mountain in the eastern townships of Quebec. We built 4 of these duckweed houses, these enclosures, plunked them down in this small little pond. And then we measured the number of fronds in the fall, right up into, they started to sink. And then we came back in the spring, as soon as the ice melted, every few days, and counted all the fronds that emerged and removed them from the enclosures. Also to control for immigration, we covered 2 of the enclosures. And then also to control for reproduction, every time we sampled in the spring, we took some fronds and pond water out of the pond into a few buckets, replicate buckets on the side, and measured rates of population growth. And here's what we find. So this is number of fronds that reemerge. Each line is a single enclosure. So fronds start to reemerge at the beginning of April. By the beginning of June, they're essentially all up. And there is, of course, a strong dependence on temperature. As soon as the water temperature hits about 10 degrees Celsius, fronds come up rapidly, sometimes as much as 5,000 individuals in just a single day in one 1 meter squared enclosure. It's pretty cool. Looking at this data a little bit differently. So we're comparing the percentage of fronds that reemerge in the spring as a function of the previous year's population in each enclosure. So we see that in all 4 enclosures, essentially close to 100% of fronds survive the winter. So there is no bottleneck. That was really surprising. It's an extremely simple study, right? It's almost a pilot study, but it teaches us a lot about the life history of Lemna minor and how they're capable of sustaining high levels of genetic diversity. So this genetic diversity is then maintained between a balance between mutation and purifying selection. So that's interspecific genetic diversity. What about species diversity? Does Lemna minor coexist with other species in the field? Does it mostly grow in monoculture? Well, I can answer that question, right? Because we did 3 weeks in the field looking for floating aquatic plants. Remember chapter 2? So we're looking at the distributions of these different species, what determines diversity, and is there ecological specialization that are responsible for the distributions of the different species. So when we were looking for duckweed in chapter 2, we found 37 different sites. We weren't just looking for Lemna minor, we were looking for any site that was suitable for any floating aquatic plants. Here they are. We found 7 species, and just by chance, in all of those different sites, Lemna minor was present. So there was no site anywhere that we found in Quebec where there were floating aquatic plants, but there wasn't Lemna minor. That's pretty interesting. The black triangles are where the species are present. When they're in red, that means that that species is also dominant in the community. So Lemna minor is everywhere and is also usually dominant. However, it's often coexisting with other species. This is just a histogram of species richness as a function. Well, yeah, histogram of species richness. And we see that Lemna minor is, yes, often in monoculture, but more often than not, it's coexisting with 2, 3, 4 other species. So our question here is what determines these different species distributions? Is there a degree of ecological specialization? So here we have the dominant species in a community plotted against the total phosphorus in the water. So I'll point out now that this is Lemna minor. So it's everywhere, all the time, usually dominant, right? So it's a best example of a generalist, a sort of Darwinian demon. But for the other species, it does seem like there is a certain amount of ecological specialization while they're dominant in different environmental conditions. And what I want to point out here is how that correlates with size, right? So the more nutrient poor, the more likely you are to have large species usually with many roots. And as the amount of water phosphorus increases, the species get smaller and smaller up until you get Wolfia columbiana. This is watermeal, another species of duckweed. It's the smallest flowering plant. The frond's about 1 millimeter. It has no roots. It's just like a ball of photosynthetic tissue. Looking at this a little bit more quantitatively, we can look at presence versus absence data. So here we have total phosphorus on both axes. And then where the species is present and where the species is absent, averaged across all 37 sites. So this 1-to-1 line is kind of like the line of neutrality, the null model showing ecological equivalency. And here's where the species fall. And it kind of maps onto an interesting taxonomic relationship as well. So here we have the larger liverworts that seem to specialize in more nutrient-poor sites, moving over to angiosperms, ceratophyllum, and then over to the duckweeds, with the smallest duckweed, Wolfia, way over here, that specialized to high nutrient environments. So species do coexist, and they possess ecological differences. There does seem to be a degree of ecological specialization. So this suggests that species sorting or competition is important in structuring these species distributions, and of course, in addition to ecological drift. So this next chapter that I'm going to present is a mesocosm experiment that is explicitly designed to measure the relative contributions of species sorting and ecological drift to community composition. We call this ABCDE, a basic community dynamics experiment. And I'm so happy this worked because we ran this over COVID. We set it up just as we got the go ahead, and it was locked down, the experiment was running fine, and then we opened back up and we could take measurements. So I'm very thankful that this one worked out. The main point of the experiment was to determine how deterministic community ecology is, right? The classic debate between niche and neutral theory. Community dynamics are governed by two opposing processes. We have species sorting on one hand, and then we have ecological drift on the other. Species sorting is natural selection at the level of the species, and it will produce a distinct assemblage of species in each type of habitat in which all the members of that community are specifically adapted to those conditions of growth, and will produce different communities in different types of habitats. This is the process that underlies niche theory, and it predicts deterministic dynamics. On the other hand, we have genetic drift. Ecological drift, which is essentially genetic drift at the level of the species, and it will result in assemblages of species in each locality, the individuals in which are completely unrelated to the local conditions, right? So it's completely stochastic. This is the process that underlies neutral theory, and it predicts, again, stochastic dynamics of species. We can imagine this basic community dynamics experiment where we have common gardens, so one set of environmental conditions, whole bunch of replicated mesocosms with two species, species A and B, so blue and red. And both species are present in all communities, but they're divided into two different community types that differ in their initial relative abundances. So you have community type A up here, where the blue species is dominant and the red species is rare. And then you have community type B, where the blue species is rare and the red one is dominant. We then allow time to pass, communities to change, we come back, see what happens. So if species sorting is a dominant process, then there will be a convergence of the different community types towards a single equilibrium community based on those environmental conditions, right? So they all start out different, but at the end, they're all the same. This will, of course, differ in different environmental conditions. If ecological drift is important, then there will be a divergence among replicate communities due to stochastic demographics. Then the third possibility is that initial state is the best predictor of a final state. This may be because communities didn't change, right? So initial equals final, or it may mean that the initial state influences sorting or drift through historical contingency, right? So you can have the initial state influencing sorting through priority effects or inhibition, or you can have the initial state influencing drift because species that start out rare are more likely to drift towards extinction, for example. The contributions then of sorting, drift, and initial state could sum to the overall change in community composition over the course of the experiment. So we ran this at LEAP, Large Experimental Array of Ponds. This is at McGill, Mount St. Hilaire. These are 1,000-liter mesocosms, replicated mesocosms outside, filled with natural lake water from the adjacent lake. So it has natural communities of phytoplankton and zooplankton. So the experiment's like what I outlined, but a little bit more complex. Now we have 4 different species of duckweed, not 2, and they're arranged into 4 different community types. Each species can either start out at 10, 20, 30, or 40% relative abundance. Each of these 4 community types are then replicated in 12 different mesocosms. So we have a total of 48 of these giant tubs. We inoculated them all with these same 4 species in these 4 different compositions at the beginning of the summer. COVID hit. Actually, that's not quite true. We prepared the experiment, COVID hit. We got the go ahead to start the experiment. We started it, and then someone was allowed to live on site and measure relative abundance every 2 weeks. And it wasn't me, so I'm extremely grateful for this, my research assistant. The main point is to partition the overall change in community composition into the contributions that reflect species sorting, ecological drift, and initial state. So how do we do that? Well, it's nice to look at this graphically. Here we have initial frequency plotted against final frequency for a single species. The blue dots are the individual mesocosms. The green dots are the mean for each community type. The red dot here is the overall grand mean for final composition. And the black circle is the grand mean for initial composition. So then within a modified ANOVA, we can partition out these sources of variation. The effect of ecological drift will essentially be just the residual variation, right? The variation among the different blue dots, among the different replicate mesocosms. The effect of initial state will be the variation among the green dots, the mean community types. And then the effect of sorting will be the overall shift in grand mean between black and red dots. So here we have the results. 4 figures, 1 for each community type. Each color is a different species. So the dynamics look relatively clean. Each line is a different mesocosm. The bold line is the mean for each species frequency in each community type. And yeah, of course, there's variation, but they're all kind of heading towards the same point. The red species is Lemna minor, surprise, surprise. It dominates every community. And regardless of the frequency at which it starts out, by the end of the experiment, it's always extremely abundant. Is it heading towards an equilibrium state, perhaps by competitive exclusion or stable species coexistence? Well, we can look at this by plotting the change in relative abundance for each species as a function of its previous relative abundance. So here we have the 4 different species. Let's just look at one. So up top, we have Lemna minor. This black line is the line of 0 change. So meaning that when a dot is above this line, Lemna minor is increasing in frequency. When a dot's beneath it, it's decreasing. So in a situation on the right, where Lemna minor is more abundant in the community, it's more likely to decrease. When it's more rare, it's more likely to increase. So this is known as negative frequency dependent selection. And it's likely an extremely important stabilizing mechanism responsible for stable species coexistence in the field. So we get these negative relationships for all species. And what's interesting is that where the regression line intersects with the line of 0 growth, this is our predicted value for the frequency of that species in the hypothetical equilibrium community. So at an equilibrium, we predict Lemna minor to make up 75% of the individuals, Spirodella to make up about 25%, and the other 2 take up the loose change. But the point of this chapter, right, was to quantify the relative contributions of these different processes. So here we have the end of the experiment, the contributions of sorting, initial state, and drift. Each color is a different species. So for purple and red, 2 of the species, we see that sorting is by far the dominant process. So red is Lemna minor, it's selected for. Purple is Wolffia, it's selected against. Whereas initial state and drift are not important. However, for the other 2 species, it's the opposite. Sorting is not important, but drift is. So what's going on here? We can take a closer look by, instead of just looking at one snapshot at the end of the experiment, by doing an identical analysis, but at every time point throughout the experiment. So here we have the 12 weeks of growth, which is about 12 generations, 1 figure per species, and then each color line is a different process. So we see that at the beginning of the experiment, initial state is the main contribution to community change. Why? Because communities haven't yet had the time to change. As the experiment progresses, the contribution of initial state starts to decay, and the contributions of sorting and drift increase. By the end of the experiment, the dynamics are mostly governed by sorting, but it's extremely species dependent. The best example is right here. We have Spirodella, where species sorting, the blue line, is marginal the entire time. So the dynamics are governed largely by ecological drift. Whereas for the other species, like especially these 2, the effect of sorting is overwhelming. So why is it that sorting is not at all important here for Spirodella? It could be that competition is just absent and that dynamics really are just drifting around stochastically, or it could be something else. Remember in the previous slide, we predicted that the equilibrium frequency for Spirodella should be about 25%, 27 exactly. Well, then just by chance, the initial frequency of all the species was 25, because the average of 10, 20, 30, and 40% of the 4 community types is 25. So it could be that there was no effective sorting because it had nowhere to go. It was already at equilibrium. So this kind of leads to a strange paradox where the more that niche theory is true, the more that species sorting is pushing species towards equilibrium, but the closer species are to equilibrium, the more that the remaining dynamics are governed by ecological drift. I'm scratching my head about that one for a while. In this chapter, we found that ecological dynamics are largely governed by species sorting, which directed communities towards a stable equilibrium maintained by negative frequency-dependent selection. But the balance between sorting and drift is species-specific. It depends on the strength of competition and on the distance a species' frequency is from its equilibrium frequency. Okay, now for the last chapter. What we're going to do is we're going to take everything that we've learned in our ecological dynamics and then add to it both evolutionary and physiological processes to see how the entire scope of eco-evolutionary dynamics will influence how the mean trait in a community will respond to environmental change. So eco-evo contributions to community trait change. The trait that we're talking about here is body size, or optimal frond area, which we've been looking at in all my chapters. Why? Well, it's extremely simple to measure. It's fundamental to the organism's physiology, ecology, and evolution. There's a huge amount of variation in frond area among the different species. There is a genetic basis to this trait, and it's also influenced by phenotypic plasticity. So the objective of this chapter is to quantify the changes in community mean phenotype in response to environmental change, and then to estimate the relative contributions of ecology, so species sorting, evolution or adaptation, and physiology, phenotypic plasticity, as well as all of the different interactions between these processes. We're talking about community mean phenotype. So this is the mean trait of species I relative multiplied by its relative abundance in the community, summed across all species. So it's essentially the average phenotype regardless of species in a community. You have species sorting or ecological change, which will influence the relative abundances of the different species. And then you have both phenotypic plasticity and adaptation, which will influence the mean value of this trait in the different species. And we're wondering how these 3 different processes will sum and interact in order to affect their overall community mean phenotype. To do this, we ran what we call a community selection experiment. So this starts with a natural source community. We sample the species from this, and ideally all the species are relatively well adapted to the environment and at a state of relative ecological and evolutionary equilibrium, ideally, right? Then we take those samples and we culture them for many generations in the original environment, but then also in a modified environment. Then at the end of this selection phase, we will assess what happened to phenotype with the use of a reciprocal transplant. So I'll just take a step back. It's a bit of a complicated experiment to describe. So of course you have this ancestral community, which should retain its traits in the original environment. Over many generations of growth in the modified environment, its characters may change, it may evolve, there may be ecological change or phenotypic plasticity, leading to a derived community at the end of this environment. So for those of you who aren't familiar with reciprocal transplants, it's a very classical and elegant way to separate phenotypic plasticity from genetic change, right? So if there's differences in species mean phenotypes at the end of the selection phase, we can then transplant them back into both environments. And if that change in phenotype is due to phenotypic plasticity, then it will reverse when you grow the plants in the opposite environment. However, if their evolutionary genetic changes, then those phenotypes will be retained regardless of which environment they're grown in. Because we're only growing the reciprocal transplant for 2 to 3 generations. So there's not enough time for evolution to act. So at the end of the reciprocal transplant, we then have a huge amount of data. We have phenotype for individuals from these 4 different assays, right? The derived and ancestral communities, assayed in both the original and modified environments. And from here, with a little bit of statistical gymnastics, we can assess the relative contributions of phenotypic plasticity, species sorting, and what am I missing? And no, not drift. So species, ecological change, evolutionary change, and physiological change. Yeah, you're right. Okay, so this is what the experiment looked like. It's at the same site as the last one, and these big mesocosms. These are a little bit smaller. They're 120 liters. We had 18 of them, which we filled up with duckweed in natural lake water with phytoplankton and zooplankton, of course. And we manipulated 2 environmental variables. We manipulated light and nutrients. So light is manipulated with the addition of these large shade cloths, different numbers of layers of them. And then nutrients is manipulated by the addition of inorganic nitrogen and phosphorus. So we have a 3x3 gradients of low, medium, and high, light, and nutrients. So we have 9 different environments. 8 of these environments are modified. 1 of them is designed to mimic the environmental conditions of the natural source population. So this is our original environment with the ancestral community. So we essentially then did this community selection experiment 8 different times for 8 different environments and then took the mean, because we're not interested in one specific set of environmental conditions. So the first phase of the experiment was in these mesocosms. This is the selection phase, 12 weeks, again, about 12 generations. At the end of that, we take samples from the mesocosms and then bring them back to the McGill greenhouse on the roof of the building. Looks very similar to yours, in these smaller tubs. And then we grow both ancestral and derived communities in both the original and modified environments for 2 weeks. It looks something like that. After 2-3 generations, to allow for the maternal and carryover effects to decay, just like a common garden, we measure phenotype, so frond area. Okay, let's take a peek at the results. So these are results just from the selection phase. So this is data taken from the mesocosms, not the reciprocal transplant. Here we have 12 weeks of growth. Each line is the mean for each set of environmental conditions. So line type is nutrients, line color is light. The black line here is the ancestral community in the original environment. And we're looking at community mean frond area. So in the original environment, frond area remained relatively constant. However, environment did have a huge impact on community mean frond area, specifically with light. So green is low light, you had large fronds, blue is medium light, medium-sized fronds, and red is high light, you had small fronds. This was due both to interspecific and intraspecific change. First, looking at interspecific change. So this is the effect of species sorting, ecology. Here we have the different competition coefficients of the different species in the 9 different environments. So each panel is a different species. So for example, let's look up here. This is the liverwort, Brachiocarpus. Really what I want to point out is that there are large differences in competitive ability among the different environments. So here, under highlight, low nutrients, highlight, low nutrients, Brachiocarpus is extremely well. Under low light, high nutrients, Brachiocarpus does not very well at all. So the more stressful in the environment, the more competitive Brachiocarpus. However, the opposite for the small wolfia, remember, more nutrients, the less light, the better it does. Important to point out here that light was so strong that light was actually stressful. So yeah, strong differences in competitive ability. So there were strong shifts in species relative abundances. Now looking at intraspecific change. So this is how did the mean phenotype of any one species change over the course of phase 1. This is both plasticity and evolution. Here's photos taken at the end of phase 1 of 3 different species. in the 9 different environments. So there's some pretty radical differences in frond area among the different environments. This is Lemna minor, it's harder to see, but with Spirodella, it's pretty striking, the difference in frond area. Wolffia is too small to photograph. Looking at that a little bit more quantitatively, here we have the 4 different species. And again, we see that mean frond area for the 4 different species diverges among the different environments. But this is all preliminary work. The main point of this experiment was to partition the contributions of the different processes with the use of, again, a modified ANOVA. ANOVA is very diverse. You can adapt it to all sorts of situations. This one was a little bit complicated because, well, I'll come to that in a second, because there's a couple confounding things and you're dealing with an unbalanced data frame. So the partition of variance can be broken into a few factors. The first is the environment. So this is the assay environment in the reciprocal transplant. It can be original or modified. This represents phenotypic plasticity. The second is the community from which the plants came. So the mesocosm environment, it can be ancestral or derived, and it combines both species sorting, ecology, and adaptation, evolution. And then the third factor is species. And we're looking at the contribution to variation as proportion of the total sums of squares. And we see that the largest contribution is that of species. This is not surprising, and it is reassuring. It means that the average differences in frond area among species is largely retained over the experiment. Wolffia didn't become larger than a liverwort. Okay, that's good. We didn't evolve like killer Wolffia fronds. Secondly, it's kind of surprising. Overall effective environment is non-existent, so phenotypic plasticity. So this is because there are strong species by environment interactions, meaning that species respond in a physiological way differently, different magnitude, but often in different directions, not just in different environments, but also for different species in the same environment. So the overall effect of environment was non-existent. Now for the complicated term, that's community. Because remember, the effect of community combines both plasticity and evolution. So we can essentially apply the price equation to the ANOVA in order to further decompose these 2 sources of variation. And we find that over just 12 generations, we have a strong and consistent effect of both ecology and evolution that are on the same order of magnitude. So the selection of genotypes and the selection of species are equally important when determining changes in community mean phenotype. However, there's also this huge interacting action between ecology and evolution. That was surprising. What that means is that ecology and evolution didn't act in the same direction. They acted in opposite directions. So in environments in which all species evolved smaller fronds, it was the larger species that had the competitive advantage, and vice versa. So it was very surprising, and we've been kind of wrestling with this for a little bit. So it's important to note that selection between genotypes and species might not always act in the same direction. So you have to be careful with generalizations. So what have we learned in this final chapter? All 3 processes, this is ecological, physiological, and evolutionary, are responsible for driving the community response to environmental change. The physiological response was species specific. The evolutionary and ecological responses were of comparable magnitude over the same time scale, but they acted in opposite directions. What this means is that we can't just extrapolate individual species adaptive or physiological responses to a community context. We have to consider selection at these different levels independently. And so with that, I'd like to thank you for your time. And of course, thank my supervisor, Graham Bell, who's intimately involved in all of this work, as well as the many research assistants that I've had the pleasure to work with, and my collaborators, Sophia Van Moorsel, who co-authored the microbiome paper, and Etienne Odekar.